Good morning everybody. So in our previous video we have talked about the different enzymes which are required during DNA replication, and in this video we will discuss about the actual process of DNA replication. So this DNA replication, it is a complex and multi-step process, and all these steps of DNA replication, though we will discuss these steps one by one, but they occur randomly. That means when the nuclease enzymes or the helicase enzyme breaks down the hydrogen bonds, at the same time the DNA polymerase adds the nucleotides on the template state. So in this way, all these steps of DNA replication, they occur randomly. So the first step of DNA replication is activation of nucleotides. That is activation of DNA nucleotides. Now, what are these nucleotides? These nucleotides are basically monomeric units of DNA polymer. And each nucleotide in a DNA, it consists of three components. The first component is pentose sugar. In case of DNA, this pentose sugar is deoxyribose sugar. The second component is a nitrogenous base, and the third component is a phosphate. So the nucleotide containing one phosphate, they are known as monophosphate nucleotides. And as we know, the DNA contains four different types of nucleotides. The first one is AMP. AMP means the adenosine monophosphate. The second is GMP, that is guanosine monophosphate. The third is CMP, that is cytidine monophosphate, and the fourth one is TMP, that is thymidine monophosphate. Now, as all these nucleotides are present in monophosphate form, so all these nucleotides are in inactivated form. So we can call them as inactivated nucleotides. And to become activate, these nucleotides have to convert into their triphosphate form, and this process is carried out by phosphorylation step, and this phosphorylation is carried out by the help of phosphorylase enzyme. So all these monophosphate containing nucleotides which are present in inactive form, they undergoes the process of phosphorylation, and this phosphorylation is carried out with the help of an enzyme that is known as phosphorylase enzyme. So this phosphorylase enzyme carries out the process of phosphorylation and converts these monophosphate containing nucleotides into the triphosphate containing nucleotides. So AMP converts into ATP, GMP converts into GTP, CMP converts into CTP, and TMP converts into TTP, that is adenosine triphosphate, guanosine triphosphate, cytidine triphosphate, and thymidine triphosphate. Now these triphosphate containing nucleotides are known as activated nucleotides. Okay, so these triphosphate containing nucleotides are known as activated nucleotides. Now, the second step of DNA replication is opening of DNA helix. As we know that DNA is present in coiled form, and for DNA replication, the two strands of DNA must be uncoiled or unbound. And this process is known as opening of DNA helix, which is brought about in three different steps. So during opening of DNA helix, first the recognition of ori occurs. That is recognition of ori. Now, this ori, it is a sequence of some specific nucleotides, or it contains ARS. ARS means that is autonomic repeating sequences, and ori is the site in our DNA from where starts the process of DNA replication. So it is a sequence of specific nucleotides which consist of autonomic repeating sequences, and from where starts the process of DNA replication. Now, in case of prokaryotes, if we talk about the prokaryotes, these prokaryotes, they contains only one ori site. They contains only one ori site, whereas the eukaryotes, they contains more than one ori site. So prokaryotes, they contains only one ori site, whereas eukaryotes contains more than one ori site. Now, in second step of opening of DNA helix, the breakdown of hydrogen bonds between nitrogenous base pairs occurs, due to which the separation of DNA strands occurs. Now, these hydrogen bonds which are present between nitrogenous base pairs, that is the 2 hydrogen bonds present between adenine and thymine, and 3 hydrogen bonds present between cytosine and guanine. So during second step of opening of DNA helix, these hydrogen bonds are broken with the help of helicase enzyme. This helicase enzyme breaks the hydrogen bonds present between the nitrogenous base pairs and results in the separation of 2 DNA strands. And each of these 2 DNA strands, now they acts as a template or parental strand for the synthesis of new and complementary strand. Now, in next step of opening of DNA helix, the removal of tension from the separated strand occurs, and this tension or the topological stress, it is removed with the help of an enzyme that is known as topoisomerase, that we have already discussed in the previous video. And the stabilization of separated strand, it is carried out with the help of another enzyme that is known as SSB, that is single strand binding protein, or it is also known as single strand stabilizing protein or enzyme. So both these enzymes, that is topoisomerase or DNA gyrase and SSB, that is single strand binding proteins, they result in the removal of tension or topological stress, that is the function of topoisomerase enzyme, and then these 2 separated strands are stabilized with the help of SSB proteins. So all these steps results in opening of DNA helix, that is the recognition of ori, the breakdown of hydrogen bonds and separation of 2 strands of DNA, and then the removal of tension and stabilization of the 2 DNA strands. Now, our next step of DNA replication is the formation of primer. Formation of primer. Now, during primer formation, now our third step of DNA replication is the primer formation, and during primer formation, the primase enzymes binds to the open DNA strands, and this primer enzyme then results in the synthesis of a RNA primer, because this primase enzyme, it is usually a DNA dependent RNA polymerase enzyme, and this primase enzyme, it binds at or in front of 3' end of template or parental strand, and this primase enzyme then synthesizes a RNA primer of near about 10 to 12 nucleotides. Now, there arises a question that though we are synthesizing a new complementary DNA strand, then why we are using this RNA primer? The reason behind using this RNA primase enzyme or RNA primer is that between DNA and RNA, the bonding occurs quickly in comparison between the two DNA strands. So that's why we are using a RNA primer instead of a DNA primer. So during primer formation, what happens? The RNA primase enzyme synthesizes a RNA primer of 10 to 12 nucleotides, and this RNA primer binds at or in front of 3' end of the parental or template strand. Similar is the case here. So here again a RNA primer is synthesized, which will consist of 10 to 12 nucleotides, and in the same way the RNA primers will be synthesized by the primase enzyme. So till now we have completed the formation of RNA primer. Our next step is elongation process. So these three steps, that is activation of nucleotides, opening of DNA helix and formation of RNA primer, they all are included in the initiation of DNA replication process. And then comes the elongation process. During this elongation process, the new complementary DNA strand will be synthesized on the parental or template strand. And this elongation process, it includes two distinct but related processes, that is leading strand synthesis on the strand having the polarity as 5' to 3' and lagging strand synthesis on the strand having the polarity as 3' to 5' or 3' to 5'. Now during elongation process, when the leading strand synthesis occurs, usually this leading strand synthesis is more straightforward of the two. During this leading strand synthesis, first of all, a RNA primase enzyme synthesizes a RNA primer of 10 to 12 nucleotides in front of 3' and this 3' and dash end of this primer, that is RNA primer, it contains a free hydroxyl group. It contains a free hydroxyl group. So the first DNA nucleotide that enters in the process, it will always bind at the 3' end of this RNA primer because it contains a free hydroxyl group or a functional group, you can say. So in the same way, this binding of nucleotide occurs on the basis of complementary base pairing, and all the nucleotides which are entering in this process, they are joined with each other in the similar fashion with the help of DNA polymerase third enzyme. So the synthesis of this leading strand, it occurs in a continuous manner in the direction of replication fork. In the direction of replication fork. So all the nucleotides which enters in the process of DNA replication, they are joined with the help of DNA polymerase third on the basis of the complementary base pairing, and it results in the synthesis of a new DNA complementary strand on the template or parental strand. So this is newly synthesized DNA strand or complementary strand, and this strand, it is the parental strand. It is the parental strand, or even you can say it is the template strand. Now because this strand, it is synthesized continuously, so it is called the leading strand. This strand is called the leading strand, or it is also known as the continuous strand because as it is synthesized in the continuous manner. Now the second strand, that is having the polarity as 3' to 5'. Okay. On this strand, the synthesis of DNA, a new DNA complementary strand, will be accomplished in small DNA fragments, and these fragments of DNA are known as Okazaki fragments. While the synthesis of this new complementary strand on this parental strand occurs in small DNA fragments, there is a reason behind it, because the synthesis of this strand occurs in a direction opposite to the direction of replication fork. As earlier we have studied in this strand, that DNA is always synthesized from 5' to 3' direction, that is. So the synthesis of this new strand occurs in the direction of replication fork, whereas the synthesis of second strand, it occurs in the direction opposite to the direction of replication fork. That's why this strand is synthesized in a small DNA fragments. So suppose if we think first the DNA is unwound up to this region, so this will be the 3 days end and this will be the 5 days end. So the unwound part have only this much of unwound DNA or uncoiled DNA. That means the primer would bind at this position. The primer would bind at this position. So this is the RNA primer, having polarity as it is 3 days. So here it will be 5 days and it will be the 3 days. Again it have a free hydroxyl group. Now the nucleotides which enters in this process, they will bind at 3 days end because it have a free hydroxyl or a functional group. So the nucleotides which enters, they will bound to this end. Now again when the DNA is unwound to this region, in second section if we think the DNA is unwound from this region to this region, then this will be the 3 days end of the DNA and here will be the RNA primer. That is 5 days to 3 days. And again the nucleotides which enters in the process, they will be bound at 3 days end of the RNA primer by DNA polymerase third enzyme and on the basis of complementary base pairing. Now again from this region, if DNA is unwound up to this part, this primase enzyme again synthesizes a RNA primer, and this RNA primer contains the 5 days end and the 3 days end, or 5 prime to 3 prime. And again the nucleotides entering in the process, they will be bound at 3 days end by DNA polymerase third enzyme on the basis of complementary base pairing. So this is the reason why the DNA on this strand is synthesized in the form of DNA fragments. So every time RNA primase enzyme comes, it synthesizes a RNA primer and then nucleotides are added on this primer. Next, then again primase enzyme synthesizes RNA primer and nucleotides are added on in the direction of 5 days to 3 days. So the direction of these fragments is 5 days to 3 days. Okay. So DNA, it is synthesized in the direction of 5 days to 3 days every time. Okay. So the reason behind the synthesis of this strand in discontinuous manner is the movement of replication fork, because the movement of replication fork is in this side and the DNA is synthesized opposite to the direction of replication fork. That's why this strand is synthesized in the form of small DNA fragments, and these fragments are known as Okazaki fragments. Okazaki fragments. And because this strand is synthesized in discontinuous manner, first this strand is synthesized, then this strand is synthesized, then this strand is synthesized. So this DNA is synthesized in the form of DNA fragment, that is in discontinuous manner, and that's why this strand, it is known as the lagging strand, or it is also known as the discontinuous strand. Discontinuous strand. The next step of DNA replication is sealing of gaps or nicks. As you know, one of the strand is synthesized in a continuous manner from 5 days to 3 days direction, and this strand synthesis is known as leading strand synthesis. But the other strand is synthesized in discontinuous manner, that is in the form of Okazaki fragments. So during sealing of gaps or nicks, the DNA polymerase first enzyme, it removes the RNA primer. It removes the RNA primer. And due to removal of RNA primer, it results in the creation of gaps between the DNA strands. So these gaps are sealed with the help of DNA polymerase third enzyme and lipase enzyme. The DNA polymerase third enzyme, adds the nucleotides on the basis of complementary base pairing, and the ligase enzyme seals these gaps. The ligase enzyme seals these gaps. So the nucleotides are added with the help of again DNA polymerase third enzyme, and these gaps are sealed with the help of ligase enzyme. In leading strand synthesis, again the DNA polymerase enzyme adds the nucleotides on the basis of complementary base pairing, and this gap it is sealed with the help of the ligase enzyme. So in this way, from two DNA strands or from a DNA helix, we have resulted in the formation of two new DNA strands. In these two new DNA strands, this strand is template, that is parental, and this strand is newly synthesized complementary strand. Same is the case here. This is the template or parental strand, and this strand is the newly synthesized complementary strand. Thanks.